Maybe we should talk about a specific organism or two. I know you wrote about Physarum, which are a slime mold, but how do organisms like this, organisms without brains, communicate within the collective and exhibit cognition?
Yeah. So, so the first thing to think about is what other types of if you don't have a brain and you don't have muscles, what kind of space can you possibly deploy cognition in? And my claim is that there are many other spaces, including metabolic state space, physiological state space, transcriptional state space, morphological or anatomical state space, and so on. And none of those things require a brain. So all cells, and in fact, in some cases, things that are not cells, navigate those kind of spaces. And some of them do it very cleverly and in very interesting ways that is not just emergence. So a lot of people like emergence and complexity, and they say, look, there are some simple rules, and from the execution of these simple rules out come complex emergent things. And this is true from fractals on up. I mean, of course, that there is such a thing as emergence, but this is much more than that. This is not just that some sort of complexity arises. What arises isn't complexity. What arises are goal-seeking agents that have competencies to reach specific goals despite things going wrong or different. And that's a very interesting thing that's beyond plain old emergence. And that happens at all the levels. So molecular networks can do this and all now.
So like a slime mold, for instance, it doesn't have a brain, it doesn't have muscles. The sorts of problems that it might have to solve are how to repair itself, how to eat, how to dispose of waste. And these are Oh, go ahead.
Well, so I'll give you an example of a couple of Physarum problems, and then let's talk about that. Then I'll tell you about a different navigational problem for a cell that I think is a better example. So Physarum. So here lots of amazing people study Physarum intelligence, and it solves all kinds of problems, including the traveling salesman problem and some other stuff. But here's what we did. You have a petri dish, six centimeter petri dish, and you put a little piece of Physarum in the center, and then you got these glass discs. These are very thin, very light glass discs. They're inert. They're just glass. There's no food on them, nothing, just glass. So what you do is you put one glass disc on one side. You put 3 on the other side. And what you'll see is that, and this is Narosha Murugan's work in my group, and you can see some videos of this attached to the paper. What the Physarum will do is, it first for a few hours, it grows symmetrically around its, that initial point. It kind of grows outwards like this, and then boom, it makes a beeline for the 3 discs. And so what it's done in the meantime, now how does it know where the 3 discs are at a distance? What it's doing all the time, if you watch it really carefully, what it's doing is it's gently pulsing and tugging on the medium that it's on. So it's on this like rubbery agar kind of medium, and it's pulling on it. And then it's reading the strain angle that comes back. And by reading that strain angle, it can get a sort of representational map of what the mass of the objects are around it. So for those few hours where it's growing evenly, what it's doing is collecting data and processing that data and figuring out, now up until that point, it hasn't actually made a decision, it hasn't gone anywhere. So what you see is these clear phases of collecting the information and then acting on that information. Now, why it's doing this, I haven't a clue. This is just one of these things that and of course there's many. I mean, they can anticipate patterned events, and they can solve a whole bunch of other kinds of problems. So now one interesting thing in Physarum is that in that case, cutting it up as an observer, as a scientist looking at it, if I decide to, if I think I have a very clear distinction between 3-dimensional space and morphogenetic space, Physarum is going to keep me humbled by saying, well, for me, those are the same thing, because my behavior is changing my shape. That's how I do behavior. So its motion through 3-dimensional space is literally by changing its shape and growing out. So that reminds us that we can't be rigid about these things. We have to change our frame depending on the organism. But I want to talk a little bit about you were talking about the wastes and stuff. This is one of my favorite examples. That we discovered a few years ago. and my student Maya Emmons-Bell and Angela Tung worked on this. You got these planaria, these flatworms. You put them in a solution of barium, and their heads explode overnight. Literally, their heads explode. And that's because barium is a non-specific potassium channel blocker. Their heads are full of neurons. The neurons are really unhappy if they can't pass potassium. So there's excitotoxicity. The whole thing blows up. Well, if you leave them in the barium, in a couple of weeks, they grow back a new head, and the new head is fine in the barium. And we were looking and saying, how is that possible? How can they survive here? And so what we did was we did a transcriptional profiling of normal heads versus barium adapted heads, right? Just to ask what's transcription? Well, what genes are different? And then of course the difference didn't have to be in the genes at all, right? So, but that's the tool we had. So we decided to look. There's only a couple of dozen genes that are different, and the kicker is, the most important part of this story is this: Planaria have never seen barium in their evolutionary history. You don't find pools of barium in the wild. So just imagine this problem. You've got I don't know, 10 to 20,000 genes. You are hit with a stressor, a physiological stressor that you've never seen before. Which of those genes am I going to up and down regulate? You don't have time to do what bacteria often do, which is to have a rare mutant that everybody else dies and the rare mutant repopulates, right? You don't have time for that. Bacteria, planaria cells don't turn over that fast. You don't have time for gradient descent because that will take too long. And also, if you turn up and down the wrong genes, you're going to kill yourself long before you find out if this is good for barium or not. So I always visualize like you're sitting, you ever seen a picture, the inside of a nuclear reactor is like buttons everywhere, right? Buttons and lights just as far as the eye could see, but low controls all over the place. And the thing's melting down. What do you do? So the ability, so now what you have here is a navigation through a high dimensional space, the space of possible gene expressions that you could have. Let's say there's, 20,000 genes. That's how many dimensions you have in your space, and you have to navigate it from where you are now, which is you're not happy, over to some ensemble of states where the right genes are just the right 12, or 15 genes are on, that's going to enable you to solve this physiological problem, which you've never seen before. So that ability to navigate this space in terms of novelty, that's the sort of thing. I mean, your body cells are doing this all the time. In fact, I think, and it's kind of weird to think about, but I think, our ability to recognize intelligence is really optimized for medium-sized objects moving at medium speeds through three-dimensional space. So all of our sense organs point outwards. We can recognize primates, crows, parrots, maybe dolphins, things like that, right? We're pretty good at that. Not great, but okay. But imagine if we evolved with a primary sense of your blood chemistry. Imagine you had like a tongue or something inside where you could constantly feel 20 different parameters of your blood chemistry. I think in that case, we would live in a high dimensional space, and we would know that we would feel internally. It's not just this; it's this high dimensional space. And I think we would have no problem recognizing our liver, our kidneys, and various other organs as intelligent agents navigating that space, because you would say, "Oh, I ate this thing, and it's new, and it's really disruptive to my physiology. But look, my body's adapted because I've taken this walk in physiological space. Amazing." So I think at that point we would have such a much better intuitive understanding of these unconventional spaces.
You're really successfully changing the way that I'm thinking about intelligence, which is great. Before I ask a more substantive question, how did you discover this about the planaria? Did you just leave them out for a few weeks? You were too lazy to clean them up or something after their heads exploded, and then you discovered that they regrew brains, or how did that happen?
No, I mean, so for a lot of these things, what you do is you do the experiment. You do an experiment. So you put them in the barium. The reason we put them in the barium in the first place was because we were trying to modify the bioelectric circuits that control the head regeneration, and so we saw that the barium was kind of toxic, and it was blowing up the heads. And we knew planaria was regenerative, and we had a discussion about the data, and we said, leave them in there because Well, who knows what it'll regenerate? We thought it would regenerate something. I will say I did not expect that it would regrow a perfectly normal head. I thought there would be some sort of regeneration. I thought it would be aberrant. I thought that maybe it would get confused and make a tail. Maybe it would make a head without neurons. I thought it would do something. So I said, leave them there and let's see. And this is one of those things that I think is really profound. Because I'll tell you something else that's I think a kind of an important implication of this. The reason that Lamarckian inheritance doesn't work, right? Let's just remind ourselves, right? So the original idea is what's associated with Lamarckism is that you're a giraffe and you keep struggling to reach the higher branches, and because of that, your offspring, right? So your genetic material gets modified, and your offspring will have higher necks, longer necks because of it. That's the original idea. The reason that doesn't work is not the reason that they commonly tell students, which is this Weissman's barrier idea that, well, your germ cells are set aside and so they can't be accessed. That's not why. We now know that it's, of course, you can there are many epigenetic kinds of processes that will affect your germline. That's not the problem. The deeper conceptual problem is that there is no gene for neck length. There's no single gene for neck length. So the point is, whether you're the giraffe or a bioengineer or whoever, if you want a longer neck, it is completely unknown what genes would you change. And development is so sort of recurrent and complex that you can't invert it. You can't run it backwards and say, oh yes, these are the genes that, right? So that doesn't work. So that's typically the reason. And until I saw this result, which was 2019? No, 2018, I think is the first time I saw this. Until I saw this result, I really thought that, no, there's a fundamental reason why strong Lamarckism doesn't work. It's because you can't tell which genes to change to improve things. But now, if it's the case that cells can solve a novel problem and figure out at least roughly, let's say out of those 20 genes that were modified, let's say only one of them really matters for being able to survive in barium, right? Because we don't know that they're all important. Maybe it's maybe it's not the minimal set, but it's a pretty small set. Okay, fine. If cells are good at figuring out which genes can resolve a physiological stressor, I mean, my God, that solves the biggest problem of this kind of inheritance. The rest is easy. Once you figure out which transcript you want, I mean, editing the genome is not hard. There are many mechanisms inside cells that can rewrite, edit DNA. That's not the hard part. The hard part is figuring out which genes are even relevant. But these cells are telling us they can figure it out. And I don't know how this is this is a single study with, actually, there's one more like it for a different drug, but we don't know how general this is. We don't know what kinds of problems it can solve. We're going to find all this out. Obviously, my lab is studying it. But if this is generic, and if cells generally have the ability to figure out what parts of their genome are the right thing to tinker with to solve a new physiological stressor, that has massive implications potentially for evolution. So that's the sort of thing. That's what I mean when focusing your attention on how cells make decisions, how they navigate these spaces, what are their competencies, what do they know, what do they measure, what do they remember. This is, I think, the key to a lot of new biology.
I'd like to continue with this example of the Planaria and returning to the framework we began with. How do you describe or think about the intelligence of the Planaria, or relate it back to this notion of the cognitive light cone and an understanding of intelligence as relating to entertaining different ways of solving problems, and thus place it somewhere on this spectrum?
Well, I only know two things for sure. The one thing I know for sure is that there are multiple levels at which you ask that question. So behaviorally, right? So they have certain competencies in behavioral assays. They have other competencies in physiology, in morphospace. Their intelligence in anatomical morphospace is sky high, because you can cut that thing into I think the record is 275 pieces. You can do some amazing things, and every piece will be able to navigate that anatomical space to get back to a perfect little worm. It's incredible. So one thing I know is that it's not just one type of intelligence. It has many different, like all of us do. It has many different types of intelligence in different spaces. And the other thing I know for sure is that we are nowhere near smart enough to recognize all of those all of those competencies. We are just scratching the surface of figuring out what these things can do.
Okay, that's very neat. So one, their intelligence in, I think you called it anatomical amorphous space, is much higher than ours, which is pretty amazing, because I think if you ask the average person on the street, is a planaria smarter than you, they're going to say in no dimension. In no dimension are they as smart as us. So that's interesting. And then also going back to this problem of binaries in intelligence, having multiple intelligences also, I can see it does not constrain research. It opens up room for research and more understanding of the planaria, especially when you don't go in to the problem already biased and thinking it's not intelligent. That just doesn't motivate you to do more research.
That's right. That's right. If you think you know which set of tools to apply, so another way to think about that spectrum of, I actually called it in my TAME paper, where I outline all this stuff. I call it the spectrum of persuadability, because what you're trying to do, it's very engineering, and TAME stands for technological approach to minds everywhere. It's a technological approach, meaning that it's all about, put yourself in the perspective of an engineer. You want to be able to predict and change the way the system functions. What tools are you going to use to do that? So in my original kind of diagram of the spectrum, there are some mechanical clocks over here, and then some thermostats, and then some animals, and then some humans, and so on. And so if you are convinced that the only tools you can bring to bear on the system, tools meaning practical techniques and concepts, right? That the only tools you're going to have are the kind that are appropriate to mechanical clocks, meaning hardware rewiring, and that includes DNA and protein engineering and all these kind of things, then that's a set of things you're going to be able to achieve. But what if your system is also amenable to the tools of cybernetics, like rewriting goal states and looking at set points of homeostatic processes? And what if your system actually is amenable to the tools of behavior science, because maybe it learns from experience, maybe it offers habituation, sensitization, associative learning, path finding, all these kinds of things. Maybe your system is even higher on the spectrum, and it can do planning, and it can do representation and counterfactual thought and language, and who knows, right? So it's all about constraining or facilitating research. Does it encourage me? Does this framework encourage me to try tools from other disciplines to see what I'm really dealing with, or does it constrain me to say this is my toolbox and it's danger? There's a real kind of teleophobia around using other tools that are normally used, let's say in behavioral science, for things that are unconventional systems, because people say, "Oh, it's a slippery slope. You can really attribute too much mind to things." I mean, yes, but the slope is much slipperier in the other direction. If you assume that you're dealing with a dumb machine when you're not, that's actually, in terms of ethics and so on, that's a much more dangerous slippery slope. So I always argue for trying to get it right. Doing empirical work to determine where your thing is on that continuum.